In this lesson, the biology of cancer, we're going to summarize all the things we've alluded to this point. That is, what are the features of a malignant cell? What traits do they have? And how do they relate to what a normal cell does? Gain of function, loss of function, oncogenes, tumor suppressors. What I want to start with, however, are two concepts. The first is Knudson's two-hit hypothesis. In the two-hit hypothesis, while studying the cancer retinoblastoma, Knudson noticed that people who were heterozygous for the deficient retinoblastoma allele developed cancer much more frequently at an earlier stage and bilaterally compared to people who are homozygous normal. What Knudson proposed then was that there must be something genetically that predisposes those patients who were heterozygous to more readily get the cancer. And that was basically how we learned about tumor suppressors and the retinoblastoma gene. It went like this. If someone were already deficient in one of the retinoblastoma alleles, it would only take one event in one cell to lead to cancer. And since all of the cells in that person's body already had lost one of the alleles, it was very likely that in the billions of cells and proliferations that we have, that one cell would lose the other one. And it didn't matter if the others didn't, because this one cell already had the first hit, the second hit would provoke cancer. Whereas, if someone were homozygous normal and were in all their cells were two good copies, it would take first the loss of one of the alleles, then the loss of the other allele in that one cell in order to provoke the cancer. This is extraordinarily unlikely. It could have been that any one of these cells had lost one of the mutations, but any one cell having one good copy wouldn't turn into cancer. So the idea was, for retinoblastoma, loss of function of a tumor suppressor required that both alleles be deleted. What this has then turned into, in a current way of thinking, is the multi-hit hypothesis. Cancer in general is not a switch ready to be flipped. A cell does not go from totally normal to totally malignant with one mutation. One mutation may set into motion the cascade of events that will result in malignancy, but it has to take an accumulation of mutations that results in one actual cancer, like a tumor. That cell that manages to accumulate all of those mutations can eventually become a cancer, but it has to accumulate multiple mutations. And that leads us to the second concept, which is the stem cell theory. That is, cancer comes from a cell that accumulates multiple mutations. The idea is that most of our cells are healthy, but with all the proliferations going on, mutations occur. And so a bunch of cells develop one of those mutations. But either they're repaired or our immune system catches them, so that reduces the population which retain the mutation. But a very few subset are able to escape all of the defense mechanisms and eventually gain all of the mutations required to be a malignancy. And that one cell then proliferates and creates the tumor. So what we're going to see here with the biology of cancer is not only the events that occur, but it has to be the accumulation of multiple mutations and the escape of multiple defense mechanisms. So that even while the first flip that gets switched may happen in many cells, the number of cells that actually that leads to a full malignancy is very, very small. And we'll also see that once the malignant cell has reached malignant transformation, it's really hard to get rid of. So what I want to do is basically talk about what a normal cell does, the signaling mechanisms involved, then whether or not to become cancer it's a gain or a loss of function, and what the malignant trait is. And we'll build a list of these mutations that a cell must accumulate. First, a cell should grow when told to, which means it should die if it's not instructed to live. This was seen in the way of growth factors. We talked about this in apoptosis. A trophic factor had to be secreted, the ligand, binding to its trophic receptor, activating AKT, turning off apoptosis. To become malignant, the cell gains the ability to become self-sufficient. Rather than receiving the stay alive signal from the cells around it, it simply goes and gives itself an autocrine signal that sustains itself. Normal cells should also die. They should die when told to. The signaling method here was, again, in apoptosis, where we talked about the death receptors. These are perforins, granzymes, fast ligand, TNF-alpha. In general, these are considered a loss of function. That is, they lose the ability to receive the signal. It can also be considered a way of gain of function if they're able to fight against the perforins, but I want you to think about this as a loss of function of the activity of the receptor. So therefore, the malignant trait associated with this is escapes immunity. Third, a cell should die when its DNA is damaged, or at least repair the damage before proliferating. This was seen in the tumor suppressors, ATM, notice the DNA damage P53 did something about it. In the low levels, it initiated high affinity receptors activating cyclin-dependent kinase inhibitors to arrest the cell cycle and initiated DNA synthesis. In high levels, it actually induced apoptosis. It's supposed to die when damaged, and these tumor suppressors mean that there's going to be definitively a loss of function. And a loss of function of tumor suppressors means that the malignant trait will be able to escape apoptosis. But worse, if a cell can't identify that it has a problem with its DNA, won't repair it, or doesn't care, what this opens up is one of the major traits of malignancy, and that this cell will now tolerate genomic instability. The DNA is not repaired, so mutations are perpetuated in each proliferation. The fact that they can't identify or don't care that their DNA is damaged means that more DNA damage will break, and more mutations will accumulate, and more opportunity for mutations will occur. In the stem cell theory, the first line of defense really is apoptosis. Self-induced, hey, I'm broken, I'm going to take myself out of commission. That probably knocks off most of the cells that are going to have malignant transformation. Then they need a growth signal, and if they don't have it because the cell doesn't need to think them alive, they die off. Our immune system, natural killer cells and the cytotoxic T cells, are surveying for cells that are doing the wrong thing, that are expressing the wrong antigen, and they induce apoptosis. So just intrinsically, through apoptosis, immune system or intrinsic activation, most of the cells that generate that first mutation simply die off. But now, once a cell has gained tolerance for genomic instability, more mutations are going to occur. Normal cells should cooperate with each other. We learn more about this in general physiology in the epithelial lecture, but signaling mechanisms like E-cadherins and beta-catenin are mechanisms that tell a cell, hey, there's no more room to grow. We saw this in wound healing. Granulation tissue came down the sides, migrated across the base, and when they made contact with each other, started to rise. The epithelial layer grew in from the sides underneath the scab and stopped proliferating once it made contact with fellow cells. There was the need for proliferation there, but also something to limit it. The loss of cell-to-cell signaling means that there will be now unregulated proliferation. If you have unregulated proliferation and tolerance of genomic instability, you're going to start generating more and more and more mutations. But even for those cells that manage to escape self-induced apoptosis or our immune system, most of the cancers that make it this far are still going to just die. And that is because every cell has a limited number of proliferations. And it's not a signal. It's simply physical. Telomeres are only a certain length, and when they get too short, the cell goes into telomere crisis. Telomeres exist at the end of chromosomes. Every time a chromosome is replicated, a few nucleotides are left off. The telomere shortens. If the telomere shortens enough, it exposes the chromosomal DNA. Exon nucleases can degrade it. The cell identifies that as a DNA break and then decides to ligate multiple chromosomes together. This is the crisis. Cells still need DNA, and as we'll learn in genetics, multiple copies of a gene do not go well for a cell. So when anaphase comes and all the chromosomes are stuck to each other, it results in a catastrophic result, and the cell dies. Telomeres probably make up most of the elimination of those cells which have accumulated these mutations thus far. But if a cell is able to have that gain of function, and it gains the ability to replace those telomeres with telomerase, the cell is said to be immortal. Because once it gains this ability, it doesn't lose it. And no longer does it have a limited number of proliferations, it can proliferate indefinitely without risk or fear of damage from the DNA because of telomeres. Now you've got unregulated proliferation, tolerance to genomic instability, and no cap on the number of proliferations. That, however, does not make a cancer. That makes a pretty damn good cancer cell. But unregulated proliferation was defined in the first lesson as neoplasia, just growth. And if even a benign lesion that's not a malignancy grows too much, it outgrows its blood supply and dies. The cells don't have any oxygen or glucose, and so the tumor necroses. Because most cells cannot grow new blood vessels. In wound healing, we saw the fibroblasts release vasculoendothelial growth factor, platelets release platelet-derived growth factor, and TNF-beta was released, which induced angiogenesis. Angiogenesis was a good thing in wound healing because it penetrated the granulation tissue and brought the needed blood supply for the very active proliferative phase. If a cancer cell gains the ability to generate new blood vessels, it can effectively grow the tumor that it becomes indefinitely. All of this is still not malignancy. All of this is just unregulated growth. The tumor can get big. What makes a cancer truly malignant is that a normal cell stays put. Even if there is unregulated proliferation and the cells don't stop growing because they touch each other, they're still contained by whatever organ they came from. to become truly malignant, there is the gain of function that allows the cancer cells to invade the tissue locally, penetrate the basement membrane, and eventually reach lymphatics and blood vessels to metastasize. And it is invasion or metastasis that defines malignancy. Remember how these angiogenesis molecules brought new blood vessels to granulation tissue. Endothelial cells from existing blood vessels were induced to proliferate and migrate. They migrated because of the ability to manipulate and degrade the extracellular matrix. Well, that same feature is going to be used for angiogenesis, and the same proteins that are able to destroy and manipulate the extracellular matrix can also be used to destroy and break the basement membrane, allowing for tissue invasion. Then, the combination of invasion, telomeres, and tolerance of genomic instability will cause the accumulation of more and more and more mutations, which will lead to the eventual dedifferentiation. Dedifferentiation is graded as anaplasia. And the more primordial the cell becomes, the less ways we have of fighting it. To review, biology of cancer is a lot of things have to go wrong. A normal cell has a lot of fallbacks that prevent cancer transformation. They need to be told to live, they can be told to die, they can remove themselves. And they've got lots of regulation in terms of their blood supply and their physical chromosomes. A cell has to accumulate all of the mutations to become malignant. The more they accumulate, the easier it becomes to accumulate more. But one cell doesn't have one event that turns it malignant. Many cells have one event that start the process. Almost all of them are taken care of by some mechanism. But if a malignancy develops, it's because one cell eked by, escaped all the mechanisms we have for preventing this, and accumulated multiple mutations which give it all of its malignant traits. And that one cell becomes the stem cell, which proliferates to become the cancer. That's the biology of cancer.